So we're going to talk about maternal descent and what that means. We remember the origins of the organelles called mitochondria and chloroplasts, which are involved in energy in eukaryotes. And we remember that they are thought to arise from a process known as endosymbiosis. That is to say that they are living inside and they have a mutually beneficial relationship with a host organism, in this case, our eukaryotic cells. And remember that chloroplasts in plants came from a second endosymbiotic event, because all plant cells, or all eukaryotic cells at that point, had mitochondria, but they did not necessarily have chloroplasts. As you see, plant cells have both. It's thought that free-living prokaryotes incorporated themselves into eukaryotic cells when a eukaryotic cell engulfed them. When we think about what happened billions of years ago, the Earth's atmosphere was reducing. It was poor in oxygen. The inside of cells is a reducing environment. At some point, cyanobacteria, the blue-green algae, they were photosynthetic, and they were creating this waste product that was extremely toxic, and it's called oxygen. And this oxygen was filling up the atmosphere. At this point, there were some aerobic prokaryotes that were capable of using this poisonous oxygen to oxidize carbohydrates, and this resulted in greater energy generation in the form of ATP. At the same time, they were able to detoxify this hazardous compound called oxygen. And at some point in endosymbiosis, the eukaryotic cells, which were developing all these organelles in terms of membranes, encountered a prokaryote that was aerobic and was capable of using this organism inside itself to detoxify this oxygen. And at the same time, the prokaryotes were capable of propagating themselves inside the eukaryote and be safe from the external environment. And there are hints as to how this endosymbiosis might be true. The endosymbiosis is thought to be true because in mitochondria and in chloroplasts, there are actually two sets of membranes, an external membrane and the internal membrane. The external membrane is identical to the membrane of the eukaryotic cell membrane, whereas the inner membrane is biochemically similar to prokaryotic membranes. There's evidence of this just by looking at the chemistry. Secondly, these prokaryotic-like organelles, they divide independently of the cell itself, and they also have their own DNA, circular DNA, like we see in prokaryotes. Additionally, these tiny prokaryotes are about the same size as free-living prokaryotes, and they also contain ribosomes that are of a prokaryotic origin. We remember that prokaryotic ribosomes are smaller than the larger eukaryotic ones. We refer to them as the 60S ribosomes in prokaryotes and the 80S in eukaryotes. With maternal inheritance, we have to remember that the sperm cell is very, very tiny. It's just a delivery mechanism of DNA, and it has a bunch of mitochondria associated with the flagellum, which helps propel it and swim to the oocyte. The oocyte at the other end is actually a very gigantic cell, and it is packed full of many different organelles, including mitochondria. So when the sperm fertilizes the egg cell, the tail or flagellum drops off, and the mitochondria are lost, meaning that we receive more genetic information from our mothers by virtue of her mitochondria. The same thing goes for chloroplasts, as we see them inside plants. So that means that all the mitochondria come from mother to offspring, and there is no paternal inheritance in that case. There are, of course, some exceptions found in nature. So they include mussels, fruit flies, and honeybees, which actually follow paternal inheritance. You can look at the link below, which I'll put into the notes, because it's a useful diagram or animation of how endosymbiosis occurred, just for review purposes. The mitochondrial chromosome is fairly small when you compare it to prokaryotes. It's structured as we see here. There are, of course, some genes associated with the normal housekeeping functions. They have 22 tRNAs. There are 13 protein encoding regions. There's a region that corresponds to ribosomal RNAs. And most importantly, because this is involved in aerobic respiration, there are enzymes associated with those functions. The cytochrome oxidases, the ATP synthase subunits, the NADH dehydrogenases. And the final thing that we're going to point out here is this region right here, called the control region, or the D loop. And it's called the control region for a very important purpose. It actually controls the replication of this. It's actually a promoter that drives these genes in transcription. So it's a very important area that's found in practically all mitochondria that have been studied. And of course, there are diseases associated with this. If you think about it, messing up mitochondria will mess up how an organism functions. And similar to what we discussed with respect to ALU insertions, there are associations with certain genes. They're not necessarily causative. But here you can see some mutations to this ribosomal RNA here result in or are associated with Parkinson's disease. Also the cytochrome B here, some dystonia, and there are some other inherited diseases that are maternally inherited, such as MRF, which stands for myoclonic epilepsy with ragged red fibers. Remember, myoclonic, that has to do something with muscles. And in these muscle cells, remember, muscle cells are packed full of mitochondria because they have to generate a lot of energy. In this case, the defective mitochondria cause what are called ragged red fibers in those muscles, and that results in this epilepsy. So we can see the structure of this chromosome is very simplistic and can result in many things. As I said, the control region, or D loop, is not an area that has genes on them. And because there are no genes in this area, there is a reduced selective pressure. That's not to say that it's not important. If you mutate this enough, bad things will happen. As I said, it's the promoter for the entire thing. But it means that this area is more free to mutation and more free of selective pressure than the other regions. And this is the area that we're going to be very interested in looking at, because you have to remember that in an organism like a human, all these genes are going to be virtually identical. But this area should be, or could be, slightly different in sequence. So this control region, or D loop, has another name. It actually has two regions within this region, and they're called hypervariable regions. There are two hypervariable regions, and in this lab, when we look at maternal inheritance, we're just going to look at one because it's easier that way. It's cheaper. Unless we were going to do some really hardcore analysis of where our mothers came from, it's not really important. We just need to look at one of these control regions. As I said, this area is about 440 base pairs that mutates frequently enough that there is diversity between individual to individual based on their tribes, whatever ethnic background they came from, or their mother came from at least. That's only for one hypervariable region, by the way, the 440 base pairs. And because it's such a small place, we can actually amplify it by PCR. And after we amplify it by PCR, we can submit this PCR amplicon, whatever's amplified. We can submit it to a sequencing facility where they will actually tell us what each base was in that amplified region of the control region. After which, we can actually then do an analysis on the sequences. We have a bunch of reference sequences in databases that we will be able to compare our sequences against, and it will, based on the homology or similarity, we should be able to see about where our mother's line came from. The haplogroup refers to clusters of alleles that are statistically linked together. That is to say that a bunch of SNPs that always go together are going to represent a haplogroup. They're clustered together. And as I said, there are two hypervariable regions, and they're often linked, so they represent a haplogroup. And because there is no recombination in the mitochondrial genome like we do in our nuclear genome, you never have this removal of mutations like we do in the nuclear genome. When we look at how humans evolved, it's thought that all humans came out of Africa in an idea called the out of Africa theory. And when we look at a map like this, you can see where humans came out of based on fossil evidence in Africa. And over time, they expanded outwards into Europe, into Asia, into Oceania, into the Americas, right? This is looking from the South Pole down. That's why it looks so odd to us, because we're used to seeing the United States right smack in the middle of the map, or at least up on the left, because that's how we read. And you can see the migration patterns that we can follow based on evidence from mitochondria. Over time, in these areas, when people settled down, there were closed breeding populations where, people within their own tribe congregated together and had offspring together. And so once there was this mutation to the mitochondrial genome, people within a certain region or tribe would propagate that mutation and would make them distinct from another tribe historically. The early diversification of mitochondrial haplogroups happened, as we see here in Africa, once again, and it's referred to as the original haplogroup is referred to as L0. And there is a metaphor that people use. They refer to the L0 ancestors as mitochondrial Eve. They use the biblical metaphor to symbolize how a common progenitor or sets of progenitors gave rise to many different ethnicities. We have to remember that this is simply a metaphor, and it doesn't refer to a single person or single ancestor. It actually corresponds to a family, multiple females, sisters and cousins that gave rise to all the different ethnicities or tribes in humanity. All subsequent haplogroups appear, and they appear to have evolved from L0. We can tell, based on the rate of mutation over time, over all these thousands of years ago, or tens of thousands of years ago, how this occurred and how people had migrated out. Once again, this is the top-down view from the North Pole, just to show you a different perspective on this map that we see here. Now, similarly, we have diversification of the Y chromosome. as I said, one of the reasons why the mitochondria is very useful is because there is no homolog for the mitochondrial chromosome. Therefore, you don't have a reversion of mutations in the control region based on homologous recombination. If we remember, in humans, there are 22 homologous pairs. If you're female, you have 23 homologous pairs because you have two X's. But men have 22 homologous pairs plus an X and a Y. Because the Y is not homologous to X, there is no recombination. The Y chromosome has hardly any useful information on it, just one single gene that does a lot of work, which tells your body to grow testes. There are some other interesting genes on there, but they're not as important. Most of it is just degraded DNA that has no function anymore. So the corresponding metaphorical individual is referred to as Y-chromosomal atom, right? Once again, there's no one single individual that gave rise to all of humanity. It was simply a matter of a family of brothers or cousins in that case. We often refer to a theoretical individual in this case, or metaphorical individual, or sets of individuals as the most recent common ancestor. Remember, it's not an actual single male. And we can see in this dendrogram here, or phylogenetic tree, how the diversification of these haplogroups also occurred in the male lineage. We often do not do testing for Y chromosomal diversification because most of us, if not all of us, are pretty sure who our mother is unless we're adopted. Whereas it's kind of a little bit difficult to determine if that is absolutely my father. More often than not, you know that's the truth. But in those cases where it's not, we don't want to get into any trouble. Also, if we were going to look at Y chromosomal diversification or lineage, we would actually have to do an STR analysis, where we would have to look at about 11 different markers along the Y chromosome to show where we came from. And secondly, it's a little bit difficult to do on the females because they don't have a Y chromosome, so this will only serve males. So why mitochondrial haplogrouping? Why do we use that instead of anything else? Well, as I said, mitochondria divide independently of cells, so there's a high copy number. There's multiple mitochondria per cell. It's often the case that you can find something, a cell, a stray cell out in the environment, and the DNA of the nucleus is degraded. And remember, in a nuclear gene, or genome, or locus, there are only two versions of that. So there's only two copies. Whereas in a mitochondria in a cell, there are many, many copies of mitochondria. So therefore, there's more copies of these mitochondrial chromosomes. Mitochondria are universal, whereas only males have Y chromosomes. That's another reason. And high copy means there's more opportunity for intact DNA to be present. An interesting case occurred recently. Here we see some molars, or teeth. And these were found in a cave in Siberia. As is the case, when you have paleontological or archaeological samples, you have very little chance to have soft tissue because those are the things that rot the fastest. These molars here came from a hominid called Denisovan man, who actually was female. But in this cave in Denisova, in Siberia, they found human remains, modern human remains, Neanderthal remains, and they found these couple of molars and one finger bone from a child. And these came from something that was distinctly not Neanderthal nor modern Cro-Magnon human. When the conditions are poor, That is to say that there's a lot of moisture around, humidity, changes in temperature, usually hot or warm temperatures, soft tissue will degrade. But in Siberia, you have this cave that was pretty much always below the freezing point of water and low humidity. The mitochondrial DNA extracted from these teeth and this finger bone showed that Denisovan man was not Neanderthal and not human. However, when you do the comparison, you find that Neanderthal differs in only 202 bases from modern humans, whereas the Denisovan differs by 385 bases to modern humans. And chimpanzees differ by 1,462 bases from modern humans. So you can see that we are very closely related to these Denisovans. We don't have a complete fossil. We don't even have a skull. We only have a couple of teeth and a finger bone. And this kind of shook things up based on the out of Africa model because of the timing. Based on molecular markers and the mutation rate, it showed that there was out of Africa movement sooner than we expected from previous fossils.